Ferredoxin-thioredoxin reductase EC 1.8.7.2. Systematic name Ferredoxin-thioredoxin disulfide oxidoreductase is a 4Fe4S protein that plays an important role in the ferredoxin-thioredoxin regulatory chain. It catalyzes the following reaction: 2 reduced ferredoxin plus thioredoxin disulfide. Displaystyle rightleftharpoons. 2 oxidized ferredoxin plus thioredoxin thiols plus 2H plus. Ferredoxin-thioredoxin reductase (FTR) converts an electron signal, photoreduced ferredoxin, to a thiol signal, reduced thioredoxin, regulating enzymes by reduction of specific disulfide groups. It catalyzes the light-dependent activation of several photosynthesis enzymes and constitutes the first historical example of a thiol disulfide exchange cascade for enzyme regulation. It is a heterodimer of subunit alpha and subunit beta. Subunit alpha is the variable subunit, and beta is the catalytic chain. The structure of the beta subunit has been determined and found to fold around the Fez cluster. Biological function: major groups of oxygen-producing photosynthetic organisms such as cyanobacteria, algae, C4, C3, and crassulacean acid metabolism, CAM. Plants use ferredoxin-thioredoxin reductase for carbon fixation regulation, FTR, as part of a greater ferredoxin-thioredoxin system. Allows plants to change their metabolism based on light intensity. Specifically, the ferredoxin-thioredoxin system controls enzymes in the Calvin cycle and pentose phosphate pathway, allowing plants to balance carbohydrate synthesis and degradation based on the availability of light. In the light, photosynthesis harnesses light energy and reduces ferredoxin. Using FTR, reduced ferredoxin then reduces thioredoxin. Thioredoxin, through thiol disulfide exchange, then activates carbohydrate synthesis enzymes such as chloroplast fructose 16 bisphosphatase, cedoheptulose bisphosphatase, and phosphorylkinase. As a result, light uses FTR to activate carbohydrate biosynthesis. In the dark, ferredoxin remains oxidized. This leaves thioredoxin inactive and allows carbohydrate breakdown to dominate metabolism. Structure: ferredoxin-thioredoxin reductase is an alpha-beta heterodimer of approximately 30 kDa. FTR structure across different plant species include a conserved catalytic beta subunit and a variable alpha subunit. The structure of FTR from Synecocystis sp. PCC 6803 has been studied in detail and resolved at 1.6a. FTR resembles a thin concave disk, 10A across the center where a 4Fe4S cluster resides. One side of the cluster center contains redox-active disulfide bonds that reduce thioredoxin while the opposite docks with reduced ferredoxin. This two-sided disk structure allows FTR to simultaneously interact with thioredoxin and ferredoxin. The variable alpha subunit has an open beta barrel structure made of 5 antiparallel beta strands. Its interaction with the catalytic subunit occurs mainly with 2 loops between beta strands. The residues in these 2 loops are mostly conserved and are thought to stabilize the 4Fe4S cluster in the catalytic subunit. Structurally, the alpha subunit is very similar to the PSAE protein, a subunit of photosystem I, though the similarity is not seen in their sequences or functions. The catalytic beta subunit has a general alpha helical structure with an 4Fe4S center. The Fez center and redox-active cysteine residues are located within the loops of these helices. cysteine 55, 74, 76, and 85 are coordinated in cubane-like geometry around the Fayes center. Enzymatic mechanism. FTR is unique among thioredoxin reductases because it uses an Fayes cluster cofactor rather than flavor proteins to reduce disulfide bonds. FTR catalysis begins with its interaction with reduced ferredoxin. This proceeds with the attraction between FTR-Lys47 and ferredoxin blue 92. One electron from ferredoxin and one electron from the Fayes center is abstracted to break FTRs. CYS87 and CYS57 disulfide bond. Create a nucleophilic CYS57 and oxidize the Fayes center from 4Fe4S 2 plus 2, 4Fe4S 3 plus. The structure of this one electron from ferredoxin intermediate is contested. Staples et al. Suggests CYS87 is coordinated to a sulfur in the Fayes center, while Dye et al. argue CYS87 is coordinated to an iron. Next, the nucleophilic CYS57, encouraged by an adjacent histidine residue, attacks a disulfide bridge on thioredoxin, creating a heterodisulfide thioredoxin intermediate. Lastly, a newly docked ferredoxin molecule delivers the final electron to the Fayes center, reducing it to its original 2+ state, reforming the CYS87, CYS57 disulfide, and fully reducing thioredoxin to 2 thiols. This article incorporates text from the public domain PFAM and Interpro IPR 004-209. This article incorporates text from the public domain PFAM and Interpro IPR 004-207. Ferredoxin, thioredoxin reductase at the U.S. National Library of Medicine. Medical subject headings. Mesh.